So in this laboratory we're going to be talking about diffusion and the special case of diffusion called osmosis, where osmosis deals with the movement of water along its concentration gradient. So, as you know from your chemistry classes, all solutes and solvents move from a concentration that is higher to one that is lower. The resulting increase in entropy makes the whole system happier, and it will do this until it reaches equilibrium. So in biology we have to deal with systems where two bodies of solution are often separated by a semi-permeable membrane, often the plasma membrane that surrounds a cell, but not in all cases. And so we're going to model that, and we're going to torture some Elodea cells to see what it does to them. So at the outset you should be aware that there are 4 different kinds of movement of solutes or molecules across the plasma membrane, and they're cartooned here. So the first is endocytosis and exocytosis, endocytosis being the process that brings molecules into the cell, and that involves forming these vesicles by using these specially coated invaginations in the membrane, and then cinching them off. They call it a purse string model, where the cytoskeletal elements just kind of tighten around the top, and it works backwards in the case of exocytosis. There's simple diffusion, which is a molecule just going along its concentration gradient, migrating through whatever it has to do so. There is facilitated diffusion, and that involves having an actual channel that is selective for a given type of molecular ion. So in essence you are catalyzing that, and that's cartooned here. So something on the outside we want to let inside the cell, so we have a special protein that binds it and brings it in. And the final one, which is the one that actually involves energy use in the form of ATP directly linked to the process. So that again uses a specialized membrane-bound molecule, but it actually uses ATP to pump your given solute in or out of the cell. So this is how we set up the concentration gradients of potassium, sodium, and calcium that are essential for the nervous system, and eliminating equilibrating those concentration gradients is what gives us the action potential that sends the nerve impulse. So it's a very important process. It's actually very quick, and also very energy intensive. Most of the energy you use in your daily life is not actually for locomotion, but rather for thinking. Just pushing those ions across the cell membrane costs you that much energy. So importantly, while the exocytosis endocytosis is not an energy-free process, the only one that is directly coupled is, for our purposes, is this active transport. So we'll do 2 things today. First, we're going to put some exemplary biological materials into some dialysis bagging, tubing, and immerse that in deionized water, and see what, if any, of those molecules can cross through that dialysis tubing, which is it's kind of cellophane plastic, and for our purposes it models the semi-permeable membrane of the plasma membrane, the cell membrane. So in this we put glucose, starch, and albumin, and of those only molecules that are small enough should get through the pores in the dialysis tubing to actually get out into this deionized water. We will then subsequently test this water for the presence of those biological molecules using Benedict's Burette and the starch test. And I'll just give it away now. The only one that's small enough to get through is the glucose, so the only thing that should be positive is the Benedict's test. And hopefully we'll see that. But one other thing will happen too, is because those solutes are concentrated in here, and because this is deionized water, the concentration of water is actually very high out here. You can think of it that way. Or you can think of the concentration of solutes as very high in here, and either way, the water wants to rush in here and dilute this concentrated solute out here, especially if those solutes can't get out into the solution, if it's not bidirectional. So what will end up happening is the water will flow into the cell, and this will swell. And that actually models what we would see in Elodia if we put deionized water on those cells. You'd see the cell swell. The converse is true if you put on a highly concentrated solution. And since we're going to do each of these, let's develop the vocabulary for it. And you should remember each of these terms and what they represent. So a hypertonic solution, hyper, very much, I don't know how else to think of it, hyper, mucho, has a lot of solutes in it. And let's just go through these first. Isotonic is essentially a balanced solution where there's no major chemical impulse for either solution to move, so it's at equilibrium. And hypotonic means it's mostly water, meaning it doesn't have very many solutes in it. I was taught, though it was confusing to me, you think of a hypodermic needle, hypo meaning beneath the skin. In that case, that means this has a low beneath tonicity, or a low concentration of solutes, though I think that's confusing. The clearer one is the hyper. So if you put a solution bearing these characteristics on a given cell, one of three things will happen. Either nothing, since we're already at an equilibrium using an isotonic solution. If you put a hypotonic solution, so a very watery one, onto a cell, the water will rush in across the semi-permeable membrane, and the cell will swell. We call that being turgid. You'll notice that the vacuole itself on the inside is actually bigger. You should be able to observe that. Conversely, if you put in a very salty solution, a hypertonic solution, essentially the water inside the cell, which has a mind of its own, will rush out of the cell and try to dilute the very salty solution out here, leaving the cell membrane peeled back from the cell wall. And we call this plasmolyzed. So all those terms are important.